We are group 13, and today we are going to talk about double positive T cell. First of all, let's check today's menu. To understand double positive T cell, understanding T cell and thymus is important. Therefore, we introduce both of them at first. Then we talk about development of T cell. Let's start with T cells. Do you know what T cells are? The vertebrate immune system contains two major types of lymphocytes, T cells and B cells. They arise from pluripotent hematopoietic stem cells in the bone marrow. T cells in the bone marrow is immature, and they must through very complicated process in the thymus. So they migrate from the bone marrow via the blood to the thymus. There are various types of T cells, and helper T cells and killer T cells are the main types. Helper T cells act as a commander by responding to antigenic stimulations and regulating the function of other immune cells. Killer T cells are involved in cellular immunity that kills and eliminates virus, infected cells, and tumor cells. As such, T cells play a critical part in immunity to foreign substances. In the explanation of T cell, the word thymus appeared a couple times. Do you know exactly what thymus is? Thymus is a fatty butterfly-shaped organ that sits in front of the heart at the base of the neck. And thymus is mainly composed of lymphoid tissue. The thymus has an outer cortex and inner medulla. There are many immature thymocytes in the cortex and more mature thymocytes in the medulla. Thymic cortical epithelial cells support T cell development.
We introduced that T cells are developed in thymus to become mature and work as lymphocytes. How do T cells develop in the thymus? Immune cells express molecules called CD antigens on their cell surfaces. CD antigen is a protocol used for the identification and investigation of cell surface molecules. The T cell development in the thymus can be divided into several stages, depending on the CD antigen, especially CD4 and CD8. Look at this sketch. T cells are made from hematopoietic precursors in the bone marrow. Hematopoietic precursors differentiate into lymphoid progenitors and move to thymus. At this point, lymphocyte progenitors have neither CD4 nor CD8. A state in which neither CD4 nor CD8 is present is called a double negative stage, and these are divided into 4 stages from DN1 to DN4. As the cell moves through the DN stage, it will then begin to express both CD4 and CD8. This state of the dual expression of CD4 and CD8 is called the double positive stage, and this cell is double positive T cell. After the DP stage, double positive T cells lose either CD4 or CD8 and become the single positive T cells. It is SP stage. Those with CD4 but no CD8 become helper T cells, and vice versa, killer T cells. As we explained, double positive T cell is one of the forms in the development of T cell that express both CD4 and CD8. and that is located in the thymus because T cell is developed to be mature in thymus. These are the references. Thank you for your attention.